Hello everyone, hope you all are doing well. So today we are going to do topic 21 of paper 2. So here we are going to discuss molecular markers and their application in plant improvement. So here we are going to discuss molecular markers and their application in plant improvement. So first of all we should know what is molecular marker. So it is a molecule contained with a sample taken from an organism or other matters. It is a molecule contained within sample taken from an organism, this is organism, or other matters It can be used to reveal certain characters about the respective source. What it can do, it can use to reveal certain characters of respective source. Suppose we are taking a sample of some from a source. So what that sample will do, it will reveal some characters of that sample, characters of that respective source. For example, let DNA is a molecular marker containing information about genetic disorders. Suppose let DNA is a molecular marker, then it will contain the information about genetic disorders, or evolutionary history, and also specific regions of DNA. So here DNA is acting as a molecular marker. So specific regions of DNA can be used for diagnosing of autosomal recessive genetic disorders or anything, identify something from DNA. This can be help in diagnosing some DNA respective related diseases, identify something related to evolution from DNA. So this is what about DNA. And also we should know that every molecular marker has some every molecular marker has some evidence of their environment, okay, where in particular location. So a molecular marker also related DNA, so it will have evidence of the environment. Suppose we are living in a tropical region, so our DNA is such and modeled ourselves for the best survival of tropical region. Suppose we go to some Arctic region or Antarctic regions, we will not be able to survive there because our DNA is not made for there. So or DNA as a molecular marker or anything, all molecular markers have presence of particular location, environment as evidence. They have some evidence of environment where it is presence in particular location. So hope you all understand it. So once in a question there was asked about short notes, write about molecular marker transgenics or some like this a question was asked. So we'll in short know what transgenics are also. What are transgenics? So this was all about molecular marker. Hope you all understand what molecular markers are. Now moving on, what are transgenics? Transgenics. So it refers to the process of introducing an exogenous, that is gene from outside, exogenous or modified gene into an recipient of same or different species of same or different species for which gene is derived. So it can be transferred into same species, it can be introduced in same species or in different species also. Okay, so it is the kind of process introducing exogenous genes from outside or modified into a recipient which requires this gene. So this was about transgenics. I hope you all understand. Now moving on, what are the applications of molecular marker in crop improvement? Applications of molecular marker in crop improvement. How molecular markers can help us in improving crops? First of all, confirmation of hybridity. Molecular markers can confirm hybridity. Confirmation of hybridity. How they confirm? By detecting zygosity, heterozygosity in F1. So here heterozygosity in F1 can be detected. They can do they can be helpful in linkage mapping, in marker assisted selection, we will discuss marker assisted selection in this topic only. Through this selection we can identify genes controlling agronomic traits and use it to improve the population. Genes with good traits can be used to improve population. We can do here trait based selection. We can also do orthologous gene mapping. We can do gene tagging, that is mapping of gene for agricultural importance. Okay, here we can map some genes which are very important for agriculture, gene of agricultural importance. We can do heterosis breeding. Example, we can do this in corn. DNA fingerprint for varietal identification. We can do DNA fingerprinting. Fingerprinting for identifying of varietal identification. For example, sex identification in dioecious plant. For example, we can see sex identification in dioecious plant. In dioecious. You all know what monoecious and dioecious plant are. So in dioecious plant we can identify sex. It can be also helpful in phylogenetic and evolutionary studies. This is number 9, phylogenetic and evolutionary studies. So this was the application of molecular marker in crop improvement. Now moving on, what is marker assisted selection? This one, marker assisted selection. Let me rub it first. Keep making notes. The notes will help you so much in this afternoon preparation. The topic I am going to teach, keep making notes of those topics. These are very much important. Most of the questions will directly from topic. Almost every question will be directly from the topics I am teaching. Now what is marker assisted selection or MAS? Marker assisted selection. It is a breeding strategy in which selection is based on markers. Here selection is based on, as the name suggests, selection is based on markers. Markers, which type of markers? Markers closely linked to the gene of interest. Obviously we will select those markers which are linked with our gene of interest. The gene which are not of our interest, why will do markers? We will select markers of those genes. Okay, gene of interest rather than gene itself. So instead of selecting the gene itself, we will select the markers that is closely related to that gene. It is indirect selection for a desired plant phenotype. Indirect selection. It is indirect selection of desired plant phenotype based on bending pattern of linked molecular markers, or you can say linked DNA or linked molecular markers. It is also known as marker added selection. Now, what is the advantage of marker assisted or marker added selection over conventional phenotype based selection? Advantage of marker assisted selection or marker added selection over conventional phenotype based selection. So first, here in marker assisted selection we use molecular markers. But here we use morphological markers. In mass we need sophisticated lab, but in conventional breeding we need a simple lab. There is less effect of environment. There is very less effect of environment or no effect of environment. Environment effect is less or not present, but almost nil. But in conventional there is very high impact of environment, very high environmental impact. Let me rub it. This is marker, this is conventional one. In marker, this is very accurate, very high accurate method, very high accurate. But in conventional breeding or conventional phenotype based selection, we see that the method is medium to high accurate. It is very high accurate. It takes short time, that is only 3 to 5 years. It takes very long time, that is around 10 to 15 years. Here we have no effect on gene interaction. But here we see that there is very high effect of gene interaction. Here QTL mapping is very much possible, but here QTPL mapping is not possible. Here screening at seedling stage for economic traits is possible. Screening at seedling stage for economic traits. It is possible in marker selection, but it is not possible in conventional one. Moving on, we see that here it is simpler process. Although it requires conventional lab, but a simpler and short process as compared to conventional process. It avoids any confusion related to environment. It avoids, as discussed earlier, confusing effect of environment. But here there is high impact of environment, and they also permit pyramiding for disease resistance. They permit pyramiding for disease resistance. While in conventional type it is very difficult to pyramid for disease resistance. Here the characters like disease resistance can be maybe confidently assayed without need of pathogen inclusion, pathogen inclusion and disease development. This is not DD, this is disease development. Disease development. They also allow accurate determination of plant genotype. Accurate determination of plant genotype. So what it does, it reduces the population sizes. So in less number of plant population we can do marker assisted selection, but for conventional selections we need a higher number of plant population to do conventional selection. They can help us in selection in off-season nurseries. We can do selection here in off-season nurseries also, but it is very much not possible in conventional types. As you see here, QTL mapping is possible here. So here it requires very much less time, space and resources. Through QTL mapping it requires less time, less resources and less space. And the last point is that here we can do negative selection. Here in MS you can do negative selection, that is we can here in negative selection we eliminate undesirable traits. In positive selection we add desirable traits, but in negative selection we eliminate undesirable, which is very much possible in marker assisted selection. So this was all about today's session. I hope you understand every concept nicely. For any queries, do comment. If you like it, please press the like button. Do share and subscribe. That's all for today. Have a nice day. Thank you.